undergrad, Diamond was a very well-rounded student. So she contributed to research and engineering projects at premier institutions around the country, even around the world. She showed her ability to be able to apply her training in engineering and mathematics to creatively solve pressing questions that address societal needs. I first had the opportunity to meet Diamond in 2021 when she visited USC after she had been accepted to the PhD program here in biomedical engineering. I was really excited to meet her in person because after reading her personal statement and her application, her motivation and her conviction for pursuing a PhD literally jumped off of the page. And so she really wanted to get this doctoral degree so that she could become an astronaut. And even beyond that, to be able to perform cancer research in space. So these are big, bold dreams, which I think characterizes who Diamond is. And she was very determined to make them a reality. This ability to think big is a characteristic that has shaped Diamond's research contributions to our lab as well. So from the time that she began working in the lab, she brought a sense of urgency and purpose in her work. She naturally thought outside of the box, which is different from most engineers, very creative person, so that she could identify creative approaches to answer questions in cancer research. And as a result, what you'll see today, she's been able to develop a foundational model for how apoptosis signaling occurs in prostate cancer cells. And this is a model that we can continue to build on in our lab. Diamond has also pursued bold goals right alongside her research. She was awarded a GEM fellowship that allowed her to complete a research internship at Lawrence Livermore National Lab. She was part of the inaugural Abracam's Leadership Development Program, where she had the opportunity to learn leadership skills that allowed her to drive change at the highest levels. And she's also the founder of Melanated Ph.D., which is a social media platform to demystify the PhD process, which we know is an important pursuit, especially in this age of misinformation and gatekeeping. So you all are in for a treat to hear Diamond present her research, and so I won't take up any more time and invite her to get started. Thank you.
Thank you for that beautiful introduction. Just before I get started, I want to acknowledge some really important guests because I know they have to leave. We have the Joan Higginbotham III, African-American female astronaut on the Zoom. So I just wanted to say thank you so much for coming and joining us. I know she has to hop off.
Thank you for having me, Diamond. I appreciate it.
Of course, of course. Good morning.
Good morning.
I'm so excited to share the culmination of my PhD research, and the title of my dissertation is Modeling the Competition Between Apoptotic and Survival Q signal responses to Optimize Anti-Cancer Strategies in Prostate Cancer. So for some context, the Q signal response is this paradigm used to describe cell signaling. And the reason why I'm interested in cell signaling is because, well, the underpin of all pathological diseases is cellular dysfunction. And so if we can understand cell signaling, then we can control cell signaling. And if we can control cell signaling, we can control cellular function. Okay. So this is way easier said than done because the cell is receiving an influx of signals at any given time. And these signals take many different forms, from autocrine signals, endocrine signals, juxtacrine signals, paracrine signals, so on and so forth. And so when we describe the cues, that is denoting these receptor binding interactions that activate biological signaling networks to regulate critical cell decisions, such as proliferation and differentiation, growth and quiescence, and then lastly, death and survival. And this latter one is where I spend the brunt of my work. Now, cell signaling, especially at the Q level, is really important because this is our first line of defense toward intercepting any of these 10 hallmarks of cancer or enabling characteristics to cancer's onset and progression. So they can range from deregulation of cellular energetics, sustained proliferative signaling, evasion of growth suppressors, induction of angiogenesis, where the tumor can literally recruit nutrients, But the one that I focus on in my work is this one right here, known as resisting cell death, which leads us to the signal. Understanding these receptor signaling interactions will enable us to force desired outcomes. And the desired outcome that we want to force in tumor cells is apoptosis. Now, apoptosis is programmed cell death. Unlike other types of cell death like necrosis, necroptosis, and even ferroptosis, which are unregulated forms of cell death. And the reason why we want to trigger apoptosis or prioritize apoptosis of the other forms is because all cells have to die at some point. So what I mean by that is when a healthy cell, ages, becomes infected, becomes injured or damaged to the point where it's no longer beneficial to be kept, appropriately, it commences this apoptotic signal. The cell shrinks, membrane blobbing occurs, and then it forms these apoptotic bodies. Now, these apoptotic bodies are still functional as they're disintegrating because the organelle function within is still conserved. However, what we know about cancer cells is apoptosis is dysfunctional, meaning when cancer cells age, become infected, or become injured, and they're no longer useful to the body, whereas they're supposed to be receiving this apoptotic signal to die, they persist. They survive, they can proliferate, and they can form these large populations of tumor cells that we know to be a solid tumor. Now, we're all familiar that we want to avoid this as much as possible because these solid tumors are what composes cancer. Now, in my work, I specifically focus on hormone-dependent cancer types because hormone-dependent cancers have unique settings that increase their propensity toward evading regulatory mechanisms, expressing receptors for growth hormones, and they can also host pro-survival signaling proteins. So what are we doing about this? So I said in the title that I wanted to optimize anti-cancer strategies. In order to do so, we have to understand that our bodies are highly fortified defense mechanisms, and they're always working in the best interest of survival, self-preservation, self-protection, and so on and so forth. And therefore, the effectiveness of cancer therapies like the ones listed here can lose that effectiveness over time once the body identifies them as foreign or as an invader or a threat. So in my work, what I'm saying is that maybe a more effective approach is to work with the body's natural defense mechanisms, i.e. apoptosis, so that we're not trying to overcome or break down this fortified wall of our body's immune system, but we're just working alongside it. And as I showed before, apoptosis is already a natural response of the body to clear unnecessary cells. So throughout my talk, I have outlined these 3 aims. And based off of this left-hand side, whatever is bolded, that's where I'm at in the discussion. And each aim has its own set of questions. In the first aim, the set of questions are set to model resistance to apoptosis itself. The second aim is meant to model resistance to pro-apoptotic treatment. And then the last aim kind of draws from insights from aim 1 and aim 2 to now no longer look at apoptosis as whether it's happening or not, but now incorporate a survival signaling pathway, the mitogen activated protein kinase pathway, to see how suppression of apoptosis can be having co-stimulatory or co-inhibitory impacts as the apoptosis signaling pathways. And all of those aims, their questions funnel into this big overarching question that I've been trying to answer over the course of my PhD, which is, what features of the Q signal response that I just described determine whether cancer cells are choosing death or whether these cancer cells are choosing survival? So to answer that question, I've developed an integrated model to understand how to overcome cancer cells' resistance to cell death. And so if we think about this conceptually, we have a population of tumor cells, and we zoom into the single cellular level, and we can investigate the apoptotic machinery to say, well, what are some vulnerabilities here that is allowing this cancer cell to evade the apoptotic signal, unlike the healthy cells? And then once we know what those vulnerabilities are, or those deficiencies are, then we can force the apoptotic signal and bring about this therapeutic benefit of these dying tumor cells, or essentially getting the tumor to eventually commit cell suicide where it's shrinking itself. And the novelty of this integrated model is that we're moving beyond whether proteins suppress apoptosis. The literature is clear about the players in the apoptotic signaling pathway, but I wanted to quantify what are the mechanisms of how suppression is executed, what is the magnitude of those individual mechanisms and the extent of its impact, which signaling routes or pathways are being engaged, what conditions or what conditions make up when suppression fails or can be disrupted, and then lastly, intervention strategies to overcome resistance to apoptosis and then later on to drug treatment. So here's a preview of the entire integrated model that I've developed over the past 5 years. And just as a preview, each aim hones in on a specific subset of the model. But we're going to start with aim 1 with the extrinsic pathway to apoptosis, so like this white shaded region. And then we're going to move to the intrinsic pathway to apoptosis. And then in aim 3, this one we're going to incorporate that MAPK pathway that's stimulated by androgens, a type of hormone. So you're probably asking, well, how do you build a computational model of biological phenomena? So the great thing about being a systems biologist and a member of the Computational Systems Biology Laboratory, shout out to CSBL, is that we prioritize this concept popularized by Aristotle that the whole is greater than the sum of its parts. So we can evaluate the relative contributions of different elements using math or using mathematical models. And specifically in my work, I use ordinary differential equations deduced using the law of mass action to simulate the protein dynamics, these post-translational modifications over time, the synthesis of the proteins, the degradation of the proteins, so on and so forth, in these in silico cells. And in silico is just another word for computerized version of a cell. Then next, we have to account for heterogeneity. Now, this is a big buzzword in biomedical research and biology in general, because this is basically the reason why we're all different, why we have different fingerprints, why you have different blood types, different phenotypes. And so we're trying to optimize anti-cancer strategies. These models have to be inclusive of a wide range of patient profiles. And so we can account for these wide range of patient profiles by varying the distribution types of these intracellular proteins. And then lastly, probably the most important step is estimating model parameters using data from previously published research. And then we can link the in vitro and in vivo experiments to the model as they're being fed into the model. And then also the models can really inform the design of the experiments themselves. And it brings about this very synergistic and complementary relationship between in silico modeling and in vivo and in vitro modeling. Now, this leads us to the first aim, to model resistance to apoptosis. So if you remember that big integrated model I showed, we actually didn't start there. We actually started with an intracellular signaling model that was already established by Ising and his coworkers. But the problem with this model is it had no extracellular arm to connect us to the extracellular matrix. So one of the things that I added were, well, not one of the things, multiple things that I added was fast ligand that is both bounded and fast ligand that's unbounded, because not all ligand is necessarily activating the intracellular system. And then I also brought in 3 key features of these binding interactions. 1, ligand receptor dissociation. 2, receptor degradation. And then the last one, the internalization coefficient. And the results of this work can be found in my first author paper, modeling the heterogeneous apoptotic response of caspase-mediated signaling in tumor cells in the Journal of Theoretical Biology. But the 2 questions that were specific to this aim were, 1, unlike healthy cells, how are tumor cells uniquely equipped to evade this apoptotic signal? And then 2, how can we force the apoptotic signal in tumor cells when they fail to do so on their own? So to answer this question, I want to describe the pathway. So I remember we started with Ising's model, and I added fast ligand and fast receptor. So when ligand binds to its receptor, it can activate caspase-3. And activated caspase 8. And activated caspase 8 can do one of 2 things. It can either trigger the activation of caspase-3, or it can form a complex with BAR, also known as bifunctional apoptotic regulator. And once caspase 3 is activated, it can either form a complex with inhibitor of apoptotic proteins, or it can commence apoptosis. So naturally, activated caspase 3 is our actuator of apoptosis. So you're going to want to keep C3A in mind. Another thing that I needed to do is define our definition of apoptotic efficacy. So the two ways that we did that is by looking at the magnitude of activated caspase 3 and the time in which that maximum level is reached for any given cell. But if you recall, the first question was, how are these tumor cells uniquely equipped? So the first thing that I did, I wish that my name wasn't covering the thing, but it's okay. The first thing that I wanted to do was vary those initial parent protein values so that we can see potentially how parent protein values can impact our apoptotic actuator, C3A. Now, these are called parent proteins because they're neither an activated species and they're not a complex species as well. So when we're varying these, what I'm saying is if we have a uniform distribution, we can randomly select a value and assign it to each of those proteins. And when we do that, we see very high variability for our actuator of apoptosis, C3A. What I mean by that is for some cells, if each of these lines is an individual cell's C3A dynamics, it's very monotonic over time, meaning it's never increasing or decreasing very much. But then we have others that have very high activation energy and have a lot of C3A magnitude, and then they decline over time. And then we have some in the middle that are kind of, plateauing. So this is helpful because we see that these initial parent proteins are showing sensitivity to our apoptotic efficacy per our definitions. But because we don't know exactly what amount of activated caspase 3 is required for apoptosis to occur, what I needed to do next was introduce arbitrary thresholds. So like I said, we're pulling from a uniform distribution for this analysis, and that just means that any of the values in this distribution are equally probable. And now I have these arbitrarily selected thresholds, a low threshold at 40,000 C3A molecules, a medium threshold at 100,000 molecules, and this high threshold in red at 160 molecules. And so we can quantify the apoptotic efficacy of the entire tumor by seeing, okay, how many lines are able to cross the low threshold, so they will be apoptotic per that threshold, or how many lines are crossing the purple threshold, so they're apoptotic per that medium threshold, and so on and so forth. And in this way, we can see the time in which these cells are becoming apoptotic, and we can see how many cells are surviving, meaning those cells that are never reaching any of the thresholds. And that's basically what this column is over here. If you can't read this, it says number of cells remaining at the end of the simulation time. We can further confirm this with the normal distribution. So where with the uniform distribution, all of these values are equally probable. For the normal distribution, those values that are in the middle have a higher likelihood of being chosen. And we see similarly that there's very high variability for the normal distribution, except unlike the uniform distribution, we have some cells that never even get to the medium or the highest threshold. And then we can also quantify how many cells are surviving at the end of the simulation time with the similar plot here, and the time, the complement of the surviving cells, and then the time in which that threshold is being reached in minutes. But then I wanted to take it a step further and understand how can we further differentiate the apoptotic cells, the ones that are crossing the threshold, and the non-apoptotic cells, the ones that are not crossing the threshold. And so back to those initial parent protein values, we did this for every single parent protein. We see that the initial number of C3 that is associated with the apoptotic group are higher levels of initial C3, which is per this lowest threshold, which is why they're both blue. And then the non-apoptotic groups tend to have lower amounts of initial C3. We're able to confirm that with this bar plots because the blue bars are more rightmost and the gray bars are more leftmost. We confirm this again with the normal distribution. We see that higher amounts of initial C3 have been associated with the apoptotic group, and the lower amounts are associated with the non-apoptotic group. So we were able to kind of answer that question. Of course, I couldn't show all the data. You can read the paper if you want. But that was one of the ways that we can answer that first question of how are tumor cells uniquely equipped to evade the apoptotic signal. And the next thing I wanted to do was see well, how can we force this apoptotic signal? How can we increase the activation energy of those cells that maybe were monotonic over time? So bringing back our thresholds, just looking at the uniform distribution at this point. What this plot is showing for the red threshold, the highest one, on the y-axis, we have the percentage of apoptotic cells, and on the x-axis, we have the time in minutes. If I'm increasing the ligand from 60 molecules to 6,000 molecules, we see slightly that the higher the ligand is, the more higher percentage of the apoptotic cells there is. Similarly, for the medium threshold, for higher amounts of ligand, we have a higher percentage of apoptotic cells. In the lowest threshold, we can really see the delineation the best. We see that for higher amounts of ligand, we see a higher percentage of apoptotic cells. And not just that, or a high magnitude of activated caspase 3, but we see a quicker time in which that activation is met with the higher ligand concentrations or amounts. Even more, remember those 3 receptor features that I added? I added in ligand receptor dissociation. Now we can see how sensitive changing that parameter is to our operational definition of the time in which that apoptotic threshold is reached. And we see that as we increase the dissociation, or how loosely it's being bound to the receptor, that we see a slower activation or slower time in which that maximum is being reached. Similarly, for receptor degradation, meaning less receptor is being available, as we increase the degradation rate, also we're seeing a slower time in which that maximum is being reached. And then lastly, for the internalization coefficient, this was not very significant over the values that we varied over, but it's still helpful to see that if we want to answer this question of how we can force the apoptotic signal, we can target the ligand, we can target the receptor, and we can also target those dissociation and degradation features. Which leads us into aim 2, to model resistance to pro-apoptotic treatment. So we're moving from just looking at how we can model resistance to apoptosis to now we're trying to model resistance to drug treatment. So the model is grown, right? We started with Ising's model just right here, and then I had fast ligand and fast receptor. And we were only looking at the extrinsic pathway to apoptosis because for type 1 cells, they largely depend on only the extrinsic pathway. But because we want these models to be as inclusive as possible, type 2 cells also depend on the intrinsic pathway. So 3 main additions have been added to the model. 1, we brought in the intrinsic pathway to apoptosis. I also brought in 3 pro-apoptotic drugs. So narciclacine, tocopherol-lactobutyrate, and celecoxib. And then now the model is specific to prostate cancer data. So whereas AIM-1 was really a generic non-tumor specific model, now we have a tumor specific deviation. I guess I could start here. So how the intrinsic model is connected to the extrinsic model. Basically, when activated caspase 3 promotes the truncation of BID, when truncated BID and BAX form, they promote what we know as mitochondrial outer membrane permeabilization. And so what is that describing? It's really just describing the mitochondria becoming perforated in a way where it can release SMAC, secondary mitochondria activated caspases, and cytochrome C. So if we follow this line, SMAC is a pro-apoptotic protein because it's inhibiting our inhibitor apoptotic proteins. If you notice, we can have IAPs that are affecting the intrinsic pathway and IAPs that are impacting the extrinsic pathway. So SMAC is inhibiting the inhibitor IAP from pulling away free C9A that will bind with IAP to form this C9A IAP complex. And if we follow how the release of cytochrome C helps to stimulate apoptosis, when cytochrome C binds to the apoptosome, this promotes the activation of caspase 9, which then further promotes the activation of caspase 3, which is our apoptotic actuator. Also, these 3 drugs that we implemented, they were all performed on the same prostate cancer cell line, but if you notice, they have different mechanisms of promoting apoptosis. Well, for narciclacine, it's blocking this dissociation, the association or binding of this complex being made. And that's helpful because the more free CAA we have available, the more stimulus we have to promote C3A activation or C3 activation. Now, the way that tocopherol-lactobutyrate promotes apoptosis is by upregulating caspase 3. So it's giving us more protein to become activated for our actuator. And then celecoxib works by, similar to SMAC, inhibiting the inhibitor of apoptotic proteins on both the intrinsic and extrinsic pathways. Any questions? Okay. And the results from this model can be found in my first author paper, Mechanistic Modeling of Intrinsic Drug Resistance in Prostate Cancer Apoptosis Signaling. Feel free to read that one too. So to move this generic apoptosis signaling model to a prostate cancer calibrated apoptosis signaling model, recall we had to use a plethora of computational tools to calibrate the model. First and foremost, we needed to use ImageJ, and ImageJ is this free software that allows you to quantify the relative protein amounts from an assay. So in this case, we use a Western blot assay. And based off of the data that we have access to, we can input those outputs in EFAS, and EFAS will allow us to measure the sensitivity of the model outputs to each of those inputs. Now from EFAS is giving us a subset of our entire parameters group that requires very high confidence in the model. And so once we have the key regulators from EFAS, that is now our new input for particle swarm optimization. And in PSO, what happens is, based off of those parameters that we got from EFAS, it's searching the parameter space for the values that have the best fit to the data based off of that objective function that we define. And so in this way, we can calibrate the apoptosis signaling model. And the two questions that I wanted to answer with AIM-2 are as follows. One, can apoptosis dynamics be accurately captured in response to prostate cancer treatment interference? And then two, which features of the model represent viable targets for overcoming treatment resistance? Or simply, why do some prostate cancer cells not respond to treatment? So we're right back at heterogeneity. We proved in the first AIM that heterogeneity had an impact, or heterogeneity of those initial parent proteins impacted the propensity toward the cell becoming apoptotic or non-apoptotic. And so it's only safe to assume also that heterogeneity can have larger adverse effects on therapeutic efficacy as well. And so how do we pinpoint exactly what is causing these range of dynamics? We can use sensitivity analysis tools. And specifically in my work, I use a global sensitivity analysis tool called EFAS, which stands for Extended Fourier Amplitude Sensitivity Testing. And using EFAS, we can quantify the impact of each parameter or input setting, those initial parent protein values, the reaction kinetics, so on and so forth, on our outputs of interest. So if you recall how we operationally define apoptotic efficacy, it was the maximum activated caspase 3 level and the time in which that was reached. Sample output of EFAS would be as follows. We can get the sensitivity index. And what this is telling us, based off of this color bar, what the outputs are most sensitive to. So because initial C3 here has a very dark purple color compared to initial bar and initial C8, we're saying that the system is very sensitive to its changes in initial C3. And not only does EFAS give us a sensitivity index score, it also gives us a total sensitivity index score. So we're not just looking at the direct effect of one parameter changing, but the total sensitivity also evaluates the combination effect or interaction with other parameters and inputs. And so as you can see, once again, it's a little bit darker for initial C3, and it's saying that the overall system, based off of its direct impact and its combination or interaction impacts with other parameters, that the system is very sensitive to changes in initial C3 relative to initial bar or initial C8, which are a lighter pink. So next, if you recall the computational workflow that I described, we have to start with the data. I mean, finding data, any modeler will tell you it can take months, usually years. I'm not going to say that. But what we wanted is not just any data sets. We needed complementary data sets. So I established this data inclusion criteria. So if we have a Western blot assay, not if we have, this is actually the one we use for tachyphylaxis butyrate. If we have an assay, the three things that had to be available across these data sets so that they could be complementary was one, they had to be implemented on the same prostate cancer cell line. So as you can see up there, PC3, they were all implemented on PC3. And then I needed the protein dynamics that were found in our model. And so as you can see, tocopherol butyrate had caspase 3 and caspase 3 cleave, which is also our activated caspase 3. And then caspase 9 and caspase 9 cleave, which is activated caspase 9. And then lastly, we needed a minimum of 3 time points. So it was a lot of data sets out there for Western blot assays, but they only gave us 1 time point. And this isn't really helpful because our model is based off of ordinary differential equations, and we're looking at the dynamics over time. So if we have only 1 time point, we don't know how it's changing. So based off of the outputs that we have from our assays, for example, C3, C3A, C9, and C9A, those are our new y-axis on our EFAS heat maps. And the x-axis is grown, right, too, because now we have all of the initial proteins from the intrinsic pathway to apoptosis, the extrinsic pathway to apoptosis, and then all of the receptor degradation and the synthesis of the proteins and all the kinetics that are involved in the model. So from here, what I did was I wanted to see how the drivers varied over drug-specific conditions. And what I mean by that is the black boxes are those that are corresponding to the untreated condition, meaning the model with no drugs turned on or activated at all. The pink bars are denoting those drivers for the tocopherol treated condition, solely by itself. Narcyclacine completely by itself in blue, and then celecoxib completely by itself in purple. And as you move to the right, you can see which drivers have the most sensitive, or which drivers are really regulating the system overall. And then from that ranking that I created, that is now our new input for particle swarm optimization. So these 10 parameters are now going to be sampled in particle swarm optimization. And if I just hone in on one example, I can create a user-defined range, a minimum in the red dashed line and a maximum, the red dashed line, and then a nominal range in this orange. And over 100 samples, basically what PSO is trying to do is search up and down to find which value gives us the closest distance to our experimental data. And so the criteria for this before we fit is it needs to be within the bounds. I mean, these would not qualify because they're hitting the bounds. And they have to have the minimum sum of squared error, so the lowest error of all of the fits. So when we do that, we're able to see how well our model is predicting the dynamics with tocopherol being fit or being treated. And what we have here is these black boxes are the experimental data points that we got from ImageJ. And the pink line is what my model is predicting for the caspase dynamics of C3A that is treated with tocopherol butyrate. And that shaded line is just the standard deviation. Similarly, for C9A, we can see how closely my model is predicting this pink line to our experimental data points. And we can do this for all 3 drugs. We can see how close narcyclacine is predicting C3 and C8A based off of the blue line, and how close they are to the black boxes for C3. And then lastly, we can do the same thing for celecoxib. And as you can see, celecoxib only had 1 protein available. So we just made that criteria of having at least 1 or more caspases available for us to fit it to. So in this way, we can answer this first question of, can apoptosis dynamics be accurately captured while showing how close our model is to the experimental data? Now, the next thing that I wanted to do was understand, well, which features of the model represent viable targets for overcoming treatment resistance. So now that we have a working model, we have a set of parameters that is as close to the data as possible. Now I'm going to explore how singly activated tocopherol butyrate, narcyclacine, and celecoxib are changing the activated caspase 3 level as we increase the drug strength. And I may say TOCO for tocopherol butyrate, NARC for narcyclacine, and Cele for celecoxib because they have long names, but just know what I'm referring to. So what we see here is that as we increase the drug for all of these drug types, that the fraction of maximum activated caspase 3 is also increasing. So the drug is working, right? But if you notice, celecoxib is much more tightly constrained than narcyclizine when it's very over the same range. So we need to understand further, well, what are some of the limiting factors that are contributing to this? So as a supplemental analysis, I now wanted to compare the untreated model to taco being activated by itself, narcyclizine or NARC being activated by itself, Sella being activated by itself, and then double combination therapy. So taco and NARC, taco and Sella, NARC and Sella, and then all three combined. And so one of the things that you quickly can tell is that taco phenyloxybutyrate seems to clearly be outperforming these other drug types, but that's not necessarily counterintuitive because they all affect the system with different mechanisms and different nodes on the pathway. But what I am interested in is what could be potentially suppressing the pronounced impact of Sella and NARC, and even with this entire triple combination therapy, that's preventing it from being an additive combination result. So I decided to return to a line of logic that you may remember from AIM-1. Remember when we were looking at the initial parent protein amounts and we were able to differentiate that the initial amounts were higher for C3 in the apoptotic group, and the lower amounts of initial C3 were corresponding to the non-apoptotic group. So I did the same thing for our combination group, meaning all of these black boxes, and the group of cells that has no treatment at all, which is all of these light gray boxes. And we've also incorporated this intrinsic parent proteins now, embed, cytochrome C, I shorten as cyto-C, secondary mitochondria activated caspase, and C9. And you see something very interesting. For C9, there's very clear delineation from its untreated population and the treated population with all three drugs. Similarly, for SMAC, there's a clear delineation. Cytochrome C has a clear delineation, BID has a clear delineation, BAR, IAP, and C8. But if you notice, I skipped C3 because something really interesting happens there. If you notice, C3 is the only group or pairing that has a shared output, shared activated caspase 3 for a group of cells that has no treatment at all, and a group of cells that has 3 treatments all acted at one time. So if you recall that question I asked of why do some prostate cancer cells not respond to treatment, these are those cells, because whether they're treated or not, they're still getting the same activated caspase 3. So this is a point of contention that I wanted to look further into. So what I want to do next is look at the conditional dependencies when I'm varying initial IAP on the x-axis, initial bar on the y-axis, and then this green bar is showing the amount of activated caspase 3. And keeping in mind that this is for a population of cells with triple combination therapy, meaning taco, NARC, and Sella all being active at the same time. And they have the same initial C3 amount. And what you see emerge very quickly is as you move to the right on this contour plot, the color of green is getting lighter and lighter and lighter and lighter. So what is that telling us? It's telling us that it's not just important that we have high initial C3, we also have to downregulate those anti-apoptotic proteins because IAP downregulation is emerging as a critical determinant of apoptotic potential, whether the drugs are there or not. So this has been identified as a limiting factor. And so that's how we can answer that second question, or one of the ways. If you want all the results, look at the paper. Which features of the model represent viable targets for overcoming treatment resistance? Now, this leads us to the last aim, which was to model apoptosis and survival crosstalk. Up until this point, AIM-1 was the extrinsic pathway to apoptosis. AIM-2 was the intrinsic pathway to apoptosis. But if you notice, we're still just in the apoptosis signaling machinery. Now, the model is fully formed, the full one that I showed you at the beginning. We have the extrinsic pathway, the intrinsic pathway, but on the far right, we have the mitogen activated protein kinase pathway stimulated by androgens and activating ERK. Now, to bring in this model, I actually used a model that was already developed in the lab by Dr. Min Song, and she was stimulating MAPK with VEGF and FGF, so vascular endothelial growth factor and fibroblast growth factor. So I exchanged that dual causal input with the androgen receptor complex. And basically how this works is once androgen, a type of hormone, binds with the androgen receptor, similar to how fast ligand is binding to the FAST receptor, it can activate RAS, RAS activates RAF, RAF then phosphorylates MEC, and then the phosphorylation of MEC leads to the phosphorylation and double phosphorylation of ERK. Now you notice that ERK has 3 ways in which it can crosstalk or have impact on the apoptotic pathway that has been confirmed with the literature, of course. So one way that ERK can impact the apoptotic pathway is that it can upregulate inhibitor of apoptotic proteins. Now, if you recall from the last aim, we don't want that because IAPs are a critical determinant of suppressing that apoptotic potential. Another way that ERK can impact apoptosis machinery is it can block the truncation of BID. And we don't want that, right? Because the truncation of BID in this complex being formed helps to perforate the membrane so that SMAC and cytochrome C can be released and then facilitate more C3A. And then ERK also impacts the apoptotic machinery by blocking the activation of our initiator caspase 9. And so we don't want that because if we have more activated caspase 3, this is helping our effector caspase 3 become activated and then subsequently commit apoptosis. So the question that I wanted to answer with aim 3 was what happens when pro-survival signaling actively has crosstalk with the apoptosis signaling model. Let me change something. So if you recall those 3 mechanisms that I described, as a first analysis, I wanted to understand the impact of those ERK inhibition terms on the target of interest. So when we vary that ERK inhibition parameter for how it's impacting TBID, we see that as we increase that parameter, TBID activity is getting lower and lower, which is what you would expect because it's blocking the truncation of BID. And then when we look at how IAP is affected by this ERK inhibition parameter, because IAP is an anti-apoptotic protein, unlike TBID, as we increase that parameter, we're seeing an increase in IAP. And then lastly, for how ERK is impacting C9A, as we increase that parameter in the model, we're seeing less and less activated caspase 9 activity because this mechanism is more closely related to the inhibition of TBID. But recall, we're interested in how this survival signaling pathway is impacting apoptosis. So it's great that we can see how they're qualitatively impacting nodes on the pathway, but now we want to see how changing these parameters are impacting our apoptotic actuator, our effector caspase 3. And what we see is that changing the ERK's parameter for TBID is not sensitive to C3A at all. In fact, all of these lines are overlapping. Now, at first I was like alarmed, but it kind of makes sense because TBID is so upstream of the pathway. It has to overcome a lot of bottlenecks before it reaches C3A. So a subsequent analysis will be seeing how we can vary those downstream proteins that are intermediary between TBID and C3A. However, for how we change IAP's parameter and its effect on C3A, there's very clear delineation. And as we increase that BID parameter, we're seeing less and less C3A. And then lastly, kind of like a middle ground, we see delineation, but not as much as our IAP inhibition term. So as we're increasing the parameter associated with blocking activation of caspase 9, we're seeing a bit less C3A as a result. However, because I'm a systems biologist, we want to know what's happening all at once. So those are just one at a time turning on those mechanisms. And now we can see how when all of these mechanisms are turned on, how is this impacting our apoptotic effector, C3A, and our initiator caspase, C9A. For C3A, we see very clear suppression, meaning that when all the ERK crosstalks are off and it's just operating parallel to survival signaling, it's all the way up here in this red line. Whereas when the ERK crosstalk is on, we see very clear suppression, a shorter disparity from the x-axis. Similarly, for activated caspase 9, when all the crosstalk is off, we're seeing this dynamic, but when we turn on all 3 of those mechanisms at the same time, we're seeing suppression of activated caspase 9. So this is showing that combined ERK crosstalk suppresses not just executioner caspase 3, but also initiator caspase 9. And so that's one of the ways that we can answer this question, and kind of like an exploratory analysis of what happens when pro-survival signaling actively has crosstalk or competition. And so In conclusion, yeah, we made it through. In summary, aim 1 was meant to model resistance to apoptosis. And what I showed you is how upstream receptor characteristics and downstream protein amounts are jointly responsible for regulating apoptotic potential. We were able to differentiate those initial parent proteins and their groups of apoptotic cells per the threshold or non-apoptotic or surviving cells per each threshold. And then we expanded the model. We went from an extrinsic model to extrinsic and intrinsic. And we showed that intracellular protein heterogeneity, particularly IAP expression, emerged quickly as a dominant constraint on apoptotic potential, even when all 3 drugs were enacted at one time. And then the last aim, we drew from the insights of aim 1 and aim 2, and now we have this prostate cancer calibrated model. But now we've incorporated this entirely new pathway, MAPK pathway, to see that while individual ERK-mediated crosstalk mechanisms each partially suppress caspase deactivation, their combined effect produces strong inhibition, revealing a heightened synergistic capacity to override apoptotic signaling. And so if the significance isn't clear, like, please care, guys. This is great work. Like, has so much potential to inform drug strategies that overcome tumor cells' resistance to apoptosis. And this model can be used to enhance the effectiveness of pro-apoptotic therapies. And then I'm really looking forward to incorporating additional pathways, because we just have fast ligand and fast receptor, but there are other death ligands. And then we just looked at the MAPK pathway, but we can bring in JAK-STAT pathway, we can bring in PI3K pathway, which are, canonical for like proliferation and metastasis. And so the model can continue to grow and grow and grow. And then probably the most important part is that this is not just confined to prostate cancer. We can use it for breast cancer. We can use it for other non-hormone dependent cancer types. And so I really look forward to doing that if Dr. Finley lets me. So future work would involve some of those things that I just mentioned, but also I would love to embed this ordinary differential equation model in an agent-based model. So actually, when I first came to USC, I was working on an agent-based model, completely different than the ODEs that I work with now. But I would love to move from the intracellular signaling to now the population level dynamics, and returning back to like the juxtacrine signals that I mentioned at the beginning, so we can see how proximity to other cells like cancer-associated fibroblasts, stromal cells, and so on and so forth can also guide clinical outcomes in terms of cell signaling, deciding cell death or cell survival. So I'm really looking forward to that. And we reached the end, and I'm going to try to get through this without crying, but I have so many great people to acknowledge. In one of my favorite books, Outliers by Malcolm Gladwell, he argues that, the success of the people that we deem as outliers is not mysterious at all. It's instead grounded in a web of advantages and inheritances and so on and so forth. And standing here, like, I can so relate to that, because I too am the product of my strong village, my community support, and I just could not do this without you all. I want to shout out the Computational Systems Biology Laboratory. We've had some great times over the years. Thank y'all for putting up with all my photo ops. Like this one, I think Nehemiah really carried this jump. I think y'all can do a little harder. I'm not gonna lie. But definitely under the leadership of Dr. Finley. I mean, she's phenomenal. She really knows. Yes. Thank you for always just pushing us to think critically and think deeper and to write clear as well. But thank you so much for listening to all of my practice talks, even no matter how bad they were, but it got us here. It got us here. And then I have to thank my village. I mean, so many of them are in the room today. I mean, I could go name by name, but I just have the best family in the world. Like, I could not have asked for a better hand of cards, and y'all don't understand how much. I'm not gonna like, moving across the country was really hard, but I'm sorry. Oh my gosh, pull it together. I'm okay. A lot of these pictures are from business when y'all came out to LA, and y'all don't know how revitalizing it was. because the PhD is so hard, but y'all really just motivated me to keep going. And then I have to thank my heartbeats. I think the PhD is hard enough. I'm so sorry. I was not planning to cry. Okay. Okay. Put it together. Put it together. Okay. Okay. The PhD is hard enough, but I think the hardest part was being across the country from my favorite people in the world. And this is the picture when we first moved to LA, and y'all had left right after this picture. And I was like, oh yeah, I'm fine. Cause I already did. Y'all dropped me off at A&T for undergrad. And, I'm grown now. I cried like a baby. I cried like a baby. Like after this picture, I was a mess. But I just want to thank my mom and dad because I'm really a product of a rich inheritance and just like so many of the blessings in their life. I think just overflow to me. And just conditioning. Like my mom used to make me say every day before school, literally since elementary school, Philippians 4:13, I can do all things through Christ who strengthens me. And also this affirmation, in matters of intellect, I, Diamond Shekinah, have a quick perception and acute penetration of retentive memory, an ardent thirst for knowledge, and the ability to express my thoughts with accuracy and precision. And when I was, okay, y'all can't cry too. But, and it's just so funny because when I was practicing this, I was like, oh, it matters of intellect, how I'm trying to have a quick perception. Like it just rolls off. And like that's one of the things that I never stuttered saying. And I said it multiple times throughout this week, but I just want to thank you guys because I'm so blessed to have you guys. And thank you for supporting me financially as well. Because but yes, that is all. And I'll take time for questions. Also have to shout out my other babies over the past 5 years, the melanated PhD, as Dr. Finley already shouted out. And also the Black Doctors Creative. Some of the co-founders are in the room today. And feel free to connect with me on LinkedIn, please, especially the committee, because I need a job. So I'm gonna need your all's connection. But thank you so much for listening, and I can take time for questions now.
You're amazing.
Awesome job.
I have a question.
Yes, please, go ahead.
The question is, what is the next research that you are interested in pursuing?
Yes, so kind of like I mentioned, I would love to apply the model to breast cancer. That's really where my motivation for this research started. My grandmother, she had breast cancer, and then also my maternal grandmother, she had 3 types of cancer. And so, as Dr. Finley mentioned in my personal statement, I talked about how I noticed early on that hormone-dependent cancer types disproportionately impact African American communities. And so, because of that, and even for prostate cancer, I don't know if I mentioned it, but Black men are 1.7 times more likely to be affected with prostate cancer than white men, and 2 times more likely to die from prostate cancer than white men. So this was something that really hit home in the community. So I would love to continue to apply this model to breast cancer and then put it in the Asian-based model, as I mentioned before, with the help of Tyler. Tyler's working on the Asian-based model, so we'll talk.
Another question.
Hey, Dad, I also have a question.
Yes, Morgan has a question. Can y'all hear?
Great talk. I'm so proud of you, first and foremost. But my question surrounds how do you think your work will influence more so in vivo and in vitro systems, actually like looking and diving within the cell?
Yeah, that's a really good question. So the great thing that I love about computational modeling is because we can circumvent a lot of the risk associated with in vivo and in vitro as well, like the high costs that are associated, the lack of clinical participants. And so it can inform those experiments because it's a way for us to simulate a wide range of values, that heterogeneity piece again, using just like high process computing. And that is automatically complementary to the in vivo and in vitro studies because it gives them a starting point, like a way to design the experiments before just doing a bunch of trial and errors over and over again. And so I think that's probably the biggest way to leverage this model, saving time and saving resources, money, and not risking actual patients as much. Does that answer your question?
So you obviously had some hypotheses in starting out, but I'm interested in what surprised you about the data.
That's a good question. What surprised me about the data? Well, one, it was really hard to find. So it wasn't, I think a lot of the times when you're trying to find data sets, there is data there. There's a lot of in vitro experimentation and so on and so forth, but it has to be complementary because you don't want to introduce bias. And I think, trying to think what surprised me. For example, when we looked at the key drivers in that chart that I created, you would expect that initial C3 will always be a leading driver because you cannot activate a protein that's not available. And remember, our apoptotic actuator was C3A. But actually, if I go back to that, plot, caspase 8 was actually a key driver. Let me see. Initial C8. So very upstream of the signal, where I was expecting for initial C3 to be a high driver since activated caspase 3 is our actuator. Any other?
Can I?
Ms. Cherie has a question. Yes.
Okay. So congratulations, Diamond. This was a great presentation, and I see that your advisor has taught you how to have that infectious energy in the work. My question is actually more philosophical. How
Okay.
have you seen yourself grow over the time of your PhD? What were the learnings that you're going to take into your next adventure?
That's a really good question, Cherie. Thank you so much. I kind of stole her as my mentor because she's Dr. Philly's mentor, but you're my mentor. She's my mentor, really. I think that I was really fragile coming into USC in the sense that I was so scared to make the wrong move. But I think the best researchers are the ones that aren't scared to take the risk. Like I mentioned with the CSBL, like they've probably heard so many practice talk, they probably can say this presentation better than me. But over time, I got more comfortable with asking questions about their research, and it gave me a more holistic picture of the field in general. And I think how I've grown is that whereas before I was so scared to ask the wrong question or ask a bad question, and now it's like I don't care about that because I'm really motivated by the results and how this can change lives. It has the potential to change lives. So I think I've evolved in my motivations. I think I've evolved in my confidence. And I think I'm way smarter than my first year here. I think so. I've learned a few things. And I think also with Dr. Finley's tutelage, she's really taught me how to think critically and outside of the box as well. I think I'm already I had a creative, angle to me, but when it comes to research, it's a different way of thinking. And I think that's a way that I've grown as well.
Perfect. Thank you.
Any other questions?
Hi, Diamond. I have a question.
Yes.
Congratulations, first and foremost, on this wonderful talk. It was really it was really wonderful to be able to attend. And my question has to do with, when we're doing this kind of computational modeling, there are a lot of, sort of assumptions you have to make. So I was wondering, looking back, on your body of work, if you were to kind of go back and sort of reassess any of the assumptions you had to make to sort of build these models, where would you begin to sort of, you think, improve the model kind of from where it is today?
That's a good question. I think one of the ways that the model could improve is more sophisticated data and more data in general. But at the beginning in the first aim, I talked about how the model was generic or not tumor specific. And when I say generic, it wasn't just completely uninformed. It was actually pulling from multiple cell types. And so it was general in the sense that it wasn't specific to one. But I think that moving forward, one of the ways that I would start would be to have a complementary data set in terms of the receptor ligand binding features coming from the same cell line, and then also the initial parent proteins also coming from the same cell line, because that was really the foundation of the work. And then also this kind of goes back to Alessi's question. I learned that because I'm designing this model, the topology is literally guiding the results as well. So one of the things that I would do differently is starting with the dynamics that we're already observing in the prostate cancer data that I had access to, using EFAS sooner so that I can know which proteins the drugs needed to be tied to in order to have the optimal effect that we would simulate in actual, clinical drug development. Does that answer your question?
It does. Thank you.
Holly. Thank you for coming. Holly is a previous CSBL member.
Yes, my question is based on just the interest I took in your results, the interest I took in how you went about expressing your dissertation very thoroughly. What has helped you humanize yourself throughout all of these many experiences that you've had to have being in touch with such a rigorous research or just such a intentional aspect of research?
Yeah. I'm like super empathetic. So I think that I was motivated throughout the entire PhD by my experience taking care of my grandmothers. And I think that my parents, watching them be caregivers, that definitely instilled in me the conviction to carry forward the research. And it was one of those things where, like, when I've been looking at numbers or like the code all day, like, this is bigger than me, like I'm doing something that impacts not just the world, but the people that I love. And so I would say that would be my answer to that. Yes, Leah. Attorney Leah.
My question is actually related to Holly's, but you mentioned sort of assumptions that went into your model. Now I'm just thinking, okay, cancer treatment is changing every day. Like there are new treatments coming out, maybe starting tomorrow, So is your model, would it be difficult to adapt it? Is it malleable? Would you have to potentially start from scratch if a new treatment comes out? Which even if you did, that's okay, because they should hire you to be also funny.
Yes.
But I'm just curious if you can say more about the difficulty of that.
No, that's a great question. And I think that speaks to the advantages of this model, because that's what I'm really interested in doing. Like, if I can have more data and more drugs, we don't just have to be confined to tocopherol, oxybutyrate, narciclacine, and celecoxib. We can now implement any of these drug types in the dynamics, because it's literally just a design choice. And Dr. Finley talks about in her Principles of System Biology class, the first chapter is about model selection and model design. So based off of the new influx of treatments, we can now apply this to the model to make sure that we're still grounded by the data itself using ImageJ and Western blot assays. So we're not just designing just whatever we want to do artistically, but we're grounded or tied back to the data itself with those computational tools like ImageJ, particle swarm optimization, and global sensitivity analysis tools. And we can also expand to other tools. We aren't just confined to EFAS, PSO, and Western blot data. We can do Southern blot data, Southern blot assays and Northern blot assays, or live cell imaging, other things.
So I have a question.
Yes.
So Diamond, Dr. Diamond, that is, your studies, your scientific studies and whatnot, I'm very impressed. Congratulations to you, first of all.
Thank you.
And I am so impressed with your work. Since I have been, monitoring in my health, my PSA numbers and things like that, and doing research and looking at seeing what I can do better. Is there have your studies allowed you to be able to, transfer any of the scientific algorithms and things that you have learned in this process to identifying holistic things that or methods that men can use specifically with the prostate health factors and whatnot?
Yeah, that's actually a really good question. So I don't want to overshoot because I'm not a medical doctor at all. But remember when I mentioned at the very beginning about how our bodies are highly fortified defense mechanisms. One of the ways that you can enhance our body's pathogen detection is lifestyle choices, healthy eating, exercise, and sleep, proper nutrition, so on and so forth. And so while I can't give any hardcore, prescription, I think that is one of the ways that we can make everyday choices that can facilitate proper immune function and physiology. Does that answer your question?
Yeah, thank you.
Thank you. Yes.
No, in the third grade, you told me you wanted to go be an astronaut.
Yeah. Yeah.
And I'm wondering from your PhD, did you figure out how that's going to connect? Because I always wondered, okay, you've never been an astronaut, and now you're learning. So I just wonder all my money has been
That's actually a really great question. That's a really great question. I think they're complimenting. I think Joan Higginbotham, I think she left. But for those of you all just came, Joan Higginbotham was the third African-American female astronaut to go into space. And she was on the call earlier, but she said she had to go. When I was in 5th grade, as Eddie said, I was really interested in science. I've always loved math. But I found out that my grandmother was diagnosed with breast cancer. And both my grandmothers were diagnosed with cancer. And Gaga's cancer was metastasized, so it was in 3 locations. And I remember panicking as a 5th grader, it's up to me. I have to cure cancer. That's literally what I have to do. And so I grew up in Maryland, the DMV area. And so I remember on a field trip, we went to the Air and Space Museum. And at the Air and Space Museum, they were talking about how all these inventions came about for initially being, developed for space travel. And so that's when the light bulb went off. I was like, if nobody can find a cure for cancer on Earth, it's in space. That was literally my logic that has carried me from then to now. And so I remember that the next day I went to school, I told my science teacher, Miss Barnes, how do I cure cancer? We literally spent recess looking up the top biomedical engineering programs in the country while everybody was out playing. And she was like, okay, you got to go to Georgia Tech or Stanford. You got to go to one of those two. And so I was really trying, or Johns Hopkins. And so that's really when I got interested in biomedical engineering itself, because my goal, I resolved in my heart, I'm going to go to space, but I'm going to space so I can cure cancer there. And I really never let go of that conviction. Later on down the line, I found out that you needed a graduate degree to be accepted into the astronaut program. So that's kind of where the PhD came in, because I needed a graduate degree. I didn't want to pay for the master's. So it was the PhD is my only other option. And then I can kill 2 birds with 1 stone by doing cancer research in the PhD. And then that would really boost my resume for the astronaut program. And so that is where it all ties in. And I think I've told you that one too many times. So I don't know why your question, your investment. But yes, that was the whole thread of logic from 5th grade to now. Yes, Shelby, Dr. Shelby almost. Saturday.
I just want to say this is not a question, but more of a comment. This is beyond Black excellence. This is beyond inspiration. And selfishly, when I met you, another Black woman getting her PhD, similar age and everything, you really inspired me and so many around you. And when I'm seeing all these data points and graphs and arrows and all these great things, and I'm like, wow, you're really doing something that we would never imagine in our neighborhoods every day, for a conversation, for a dialogue, for a coffee chat. And I just want to say that publicly, as we're in a room with multicultural, different ethnicities. This is not just the average PhD. This is not seen every day in the Black community, let alone every second, every 2 months, every year. Let's take it further. This is not seen at all. So for you to be a blueprint, for you to be a staple, for you to be impactful, and to have a leader of representation who looks like you, be your leader, advisor, this is beyond legacy at this point. This is a generational formation that is going to change the trajectory in the way that things are seen and what Jesus allows it to do. And so I'm just so grateful for you. And you have inspired me to be confident on Saturday in my defense. And I'm just grateful for you.
Thanks for sharing.